Hello, everyone, and welcome to our webinar and discussion on GMOs are a failed technology. The future is GMO free. We are very honored to have with us today Dr. Vandana Shiva, founder and president of Navdanya and Navdanya International, Chito Medina, founding member of MASIPAG, Farmers, Scientists, Partners for Development, and former national coordinator of the network. He's now an associate professor of environmental science in a leading university in the Philippines. Adelita San Vicente, Doctor in Agroecology, Director General of the Primary Sector and Natural Resources in Mexico. Farida Akhtar, Founding Executive Director of UBIC Bangladesh. And Jonathan Latham, Molecular Biologist and former genetic engineer, now editor of Independent Science News. And I'm Ruchi Shroff, Director of Navdanya International and moderator today. Thank you everyone for being here. We have been repeatedly told over so many decades that genetically engineered crops will save the world by increasing yields and produce more food. They will save the world by controlling pests, they'll control weeds, they will save us by reducing chemical use in agriculture, and they will provide the resilience in times of climate change. They will save our health and malnutrition crisis through crops like golden rice and GMO banana. But none of these promises or these miracles promised by these technologies have actually proven to be true. We know that GMOs are part of an industrial agricultural system that is destroying the planet, it is depleting our water resources, it is increasing greenhouse gases, it is destroying biodiversity, and farmers and communities are losing access to the food systems that have sustained us throughout time. So citizens and farmers and movements can see through these false promises and failures of GMOs. And over the last so many years and decades, they have pushed governments to strictly regulate and ban GMOs in many countries throughout the world. But despite the known failures and the negative health and environmental effects of these GMOs, and to circumvent these regulations and bans, the industrial agricultural lobby has now started to push for a second generation of GMOs, which they call new breeding techniques, especially after countries, for example, in Europe, impose strict rules on the first generations of GMOs, and multiple countries ban GMOs this year. We are also seeing at the same time, despite all the efforts of takeover of our food system by these lobbies, the resistance in countries such as Mexico, Italy, Peru, Philippines, Bangladesh, India. And we will hear about this too today. So to start with, I would like to invite Chito Medina in Philippines to tell us about what is happening in Philippines on the ground, especially with regards to the imposition of golden rice, and what is the campaign and the resistance to the imposition of golden rice and GMO. We spoke about in our last webinar a couple of months ago, how the Bill and Melinda Gates Foundation is, along with the Syngenta and other private companies, are behind the funding of golden rice. Could you take us through what's exactly happening in Philippines right now? Over to you, Chito.
Can you see my slides?
Yes, we can. Yes, we can.
Okay. So today I will be sharing the corporate push, as well as, on the other hand, the civil society opposition of golden rice in the Philippines, specifically on golden rice. This golden rice actually is a long history. It started in 1991 to 2000, so it took about 30 years now in the development of this product. And it was very low, with the composition of only 1.6 microgram per gram of beta-carotene. And the expenses has been about 130 million US dollars, plus some additional more than almost $30 million from Bill and Melinda Gates Foundation. So the expenses is almost about 160 million US dollars in the development of this one particular GMO product, which is golden rice. The first golden rice, GR1, was in 2004. It's still very low composition of 8 microgram per gram beta-carotene. By the way, the initial one, 1.6 microgram per gram, a person needs to eat something like 9 kilogram of rice in order to get the vitamin A in this golden rice, which is very low composition. Where there were developments in 2005, they have managed to increase the numbers of the concentration of beta carotene. But again, when they cross it into commonly grown rice varieties in Bangladesh as well as in the Philippines, it has gone to an average of 3.57 microgram per gram again, which is very low. In the Philippines, the confined field trials was started in 2011. PhilRice is the Philippine Rice Research Institute. And 2012 and 2013, it was uprooted in the Philippines. This is a picture of the uprooting of golden rice by civil society organizations, consumers, and farmers in the experimental station of golden rice in 2013. 2014, the International Rice Research Institute declared that the yield of golden rice is very low, so they have conceded and they tried to improve it. So about a couple of years or 3 years, they have developed something better. So that was the developments in the Philippines. But now, actually, what is moving forward the developments of golden rice in the research institutions, in the Philippine Rice Research Institute in the Philippines and the Bangladesh Rice Research Institute in Bangladesh. The Bill and Melinda Gates Foundation has provided 10.2 million US dollars for a period of 7 years, and then extended again for another 18 million US dollars for the same countries, Bangladesh and the Philippines, to develop and to commercialize the golden rice. So we expect actually that by 2022 or in about a year, the golden rice will be approved already by governments, particularly in the Philippines and in Bangladesh. So in the Philippines, the next steps are something like this. Well, this has been done already in December 2019. Actually, our national government has approved direct use for food, feed, and processing, December 10, 2019. And therefore, if it is approved for direct use for food, feed, and processing, the regulatory process as a GMO is already over as a GMO because it can be directly consumed for food, feed, and for processing. But what they're now doing is agronomic characterization. They are doing multi-location testing in 7 locations for varietal registration. And perhaps this is a space for them to be trying to test what is the current climate of opposition of consumers. They said there is a feeding test, but actually this is not a regulatory testing. It's just a voluntary feeding test. And the feeding test is for for curing vitamin A. It's not for toxicological testing. And then they will be commercially releasing it. Let me give you a flashback. This is a study in India showing the abnormalities of the golden rice. In this case, the shorter panicles and the dwarf plants here are the golden rice, GMO plants, and therefore they are abnormal here. I think one of the explanations of the scientists is that the chlorophyll pigments had been severely affected, that the photosynthesis had been reduced and creating ultimately short panicles of the golden rice in India. In Bangladesh, the findings of the Bangladesh Rice Research Institute is only about 10 microgram per gram beta carotene average. And this is actually a few years now, and there is complete blackout of data as to what is now going on in Bangladesh Rice Research Institute with respect to golden rice. Well, there are many concerns on golden rice. I don't have to elaborate them perhaps, but we would like to mentioned that these are not pre-clinically tested to animals, and now they're saying that it can be directly used for food, feed, and processing. So this is a problematic from the point of view of safety issues. Some studies done by those uh, scientists below here, these are published in referred journal, the degradation of beta carotene in storage for 6 months is about 80 to 84% at 25 degrees centigrade. And it can change, the degradation can be lower if it is in vacuum packed, but this is not practical under farmers' condition. And in cooking, the beta carotene is further reduced by about 24%. In that case, if you look at the reality of the advantage of golden rice, a fresh golden rice in the Philippines could be 3.57 microgram per gram. Reduce, if it is stored by 3 months, then it would be about 40% reduction in beta carotene. So the amount would only be about 2.14 microgram per gram beta carotene in the golden rice. And after cooking, which is 24% reduction in beta carotene, so the ultimate beta carotene is only 1.63 microgram per gram when it is cooked and stored for 3 months. Now, no one has studied yet about this, how much is absorbed of this 1.63 microgram per gram beta carotene if there is no fats into the diet of poor people. And therefore, it implies that the benefits would be very minimal, negligible. Now, if people would like to ask how much is needed, retinol activity equivalent for children is about 250 RAE. For adults, it's 500 to 700. But the data available is something like 12 beta carotene units is equivalent to 1 retinol. But some, the pro-GMO are using something like 6 conversion factor. On the average in the Philippine context, a person eats about 0.33 kilogram per capita per day uncooked. And when you cook, the expansion is something like 2.4 units, so 0.8 kilogram cooked rice per day. I'm showing you this because then how much is needed to eat. If a single child would like to really get all the vitamin A, then the child must be eating something like 4.4 kilograms or 4,400 grams cooked rice per day in order to be nourished with the vitamin A from the golden rice. Or if they eat only the average volume of rice, the vitamin A taken from that golden rice would only be supplying 18% of the needs of the child, very small, very small. Now, the pro-GMOs had been trying to go around and they have gone to apply for approval in many non-rice eating countries in Australia, New Zealand. They're not major rice eating countries, as well as in Canada and in the United States. In the United States, of course, we all know that approval is by means of deregulation, so that the Food and Drug Administration has been saying no further question to ERI, therefore it is tantamount to being approved. So it's not a approval because the safety data has been satisfactory. Rather, there is no question, therefore it is approved. So you can see this is actually the substantial equivalent parallel idea. In fact, the letter of the Food and Drug Administration has said explicitly that golden rice does not meet the nutritional requirements to make a health claim, as the concentration of beta carotene is too low. So, if that is the case, what is the real objective of pushing golden rice by the corporations? Actually, it is to soften resistance against GMOs and pave the way for the acceptability of GMOs in the long term. It will lead, of course, to the control of seed, agriculture, food, and as well as food by the biotech companies through the GMO seeds, as well as patenting of such seeds. So the strategies of the pro-GMO corporate lobby is that the golden rice has recasted an image of biotech corporations as philanthropic and humanitarian. They created the Golden Rice Humanitarian Board to show that they are really for philanthropy and humanitarian. Because, as mentioned by Ruchi earlier, there was no successful GMO so far. They're all obviously for the betterment of or for the interest of biotech corporations. They had been arguing also that the pro-GMO lobbyists had been saying that stopping golden rice is a crime against humanity. I would argue that the biotech companies, on the other hand, should be the ones charged of crime against humanity for contamination and erosion of biodiversity, for making rice, which is the staple food of majority of global population, into a risky and potentially toxic food. Also, the biotech companies are selling the seeds and food without adequate safety testing, and therefore they are the ones committing crime against humanity by sabotaging the food, sabotaging agriculture, and sabotaging nature. The actually players, if you want to unmask the players of golden rice, initially Syngenta was the owner of this, but they said they have given it for free for humanitarian reasons. Now, Bill and Melinda Gates Foundation is the one supporting the research and up to commercialization, and he has some vested interest in GMOs. And ERI, they're using ERI to project an image of neutral research for the development of golden rice, and they have been using the Philippine Rice Research Institute, or PhilRice, to appear as a national research. So you can see the hidden agenda and the front organizations working for the interest of biotech corporations. In the Philippines, I already have mentioned that the biosafety permit for direct use for food feeding processing has been approved. The field testing also has been approved. And as I said already earlier on, we expect to be released the golden rice possibly next year. So this is an illustration of the pro-GMO advocacy. So they are now already here, field trials for varietal registration. The regulatory safety assessment, but it's not safety assessment, has been over. Some marketing tests and nutritional study, but these are not regulatory requirement anymore, and therefore they can always release golden rice anytime they want by now. Of course, we know that there are practical strategies to combat vitamin A deficiency. I don't have to mention them because of the lack of time. I would like to mention the diversity, especially we are in the tropics, that so many food has very high level of beta carotene. The reference point of golden rice in the Philippines, 3.57 microgram per gram, and you have 19 times the concentration of beta carotene here for moringa, for example, or Indian drumstick is 67.8 microgram per gram, or 19 times to that of 3.57. So you see, the red numbers here in parentheses are the times 33 units or times as much as the golden rice. So no need for golden rice. That's what I would like to say. This is another data of still many food that are much higher than the golden rice in terms of beta carotene content. So now there is no study yet as to the availability or efficacy of golden rice, but there is a study already published in referred journal that beta carotene rich orange plus potato can address the deficiency of golden rice in children. So why is golden rice pushed so hard? I think I mentioned this already, to soften resistance against GMOs and pave the way for the acceptability of GMOs in this long term. Oh, I'm sorry, this is a repetition. So what shall we do now in the Philippines? we are now at the stage where consumers should be the active opponents of golden rice, GR, golden rice. We need to develop strategies to counter the mass production of golden rice. We need to organize, develop, support, and local groups in opposing golden rice. We lobby the local government units to make ordinances banning golden rice. We need to stop legally in the courts the golden rice now. In the Philippines, there is the writ of environment, kalikasan is environment. And we have actually Stop Golden Rice Network for Asia, as well as Stop Golden Rice in the Philippines. And we would like to make this actively doing a multi-pronged campaign in order to stop really the release and commercialization of golden rice. Finally, I would like to say that, of course, our campaign is no to golden rice. Thank you very much. Thank you, Ruchi.
Thank you, Chito. Thank you very much for not only unmasking the complete false solutions behind these technologies such as golden rice, which not only do not resolve any of the problems that they have created in the first place, but we also know that the solutions to these malnutrition and health crises lie in our biodiversity, which needs to remain in the hands of the farmers and the hands of women. Thank you, Chito, for also unmasking the players behind. We have talked about it in our report, Gates to a Golden Empire, Gates to a Global Empire, on how the Bill and Melinda Gates Foundation and a convergence of multiple players are behind a lot of these techno fixes being pushed throughout our different aspects of food and agricultural system. I now go to Farida ji, who in Bangladesh, with many movements, have been battling the imposition of BT eggplant or BT Brinjal, which we also know has been failing, but there is once again a huge lobby of agribusiness and philanthrocapitalists that is imposing BT eggplant in Bangladesh. In India, many movements and farmers and networks fought against the imposition of BT Brinjal and were able to get a moratorium against BT Brinjal. And after that, we have been seeing a relentless effort by various lobbies to impose BT eggplant in Bangladesh. Could you tell us, Farida ji, what is happening in Bangladesh? Thank you.
Thank you, Ruchi, for inviting me to speak today. Can you hear me?
Yes, we do.
Yes. Okay. So, you know, we heard Chito speak about golden rice, and now I'm going to talk about, we call it BT brinjal, and it is BT aubergine or BT eggplant. But here I have nothing to give you as scientific anything, you know, it's already a game of failures and corporate lies. So what I'm going to present today is about the failure of the BT brinjal and the corporate lies. So before that, I proudly want to say, as a South Asian and also as a Bangladeshi, that we are among the few known countries as country of origin of Melungena, and Bangladesh, according to Bangladesh own government records, we have 248 different varieties and cultivars of brinjal. You know, so I can just show you, you know, different varieties. These are all growing in the farmers' fields, and they are available everywhere in different districts, different villages, you know, and also what BT Brinjal claims that it is trying to resist against the fruit and shoot borer. Already the indigenous traditional varieties, there are 24 brinjal varieties according to the Bangladesh Agricultural Research Institute, which are already resistant to this pest. So we actually did not need BT brinjal. We have our own varieties. So just some facts about brinjal is that it is very important vegetable crop in our country. It grows in 2 seasons, and all these small scale farmers are growing in their fields and even in their kitchen gardens. And but Bangladesh Agriculture Research Institute has been developing high yielding varieties and hybrid for Rabi season, means the winter season crop. Private seed companies are also coming up with hybrid varieties, and commercial brinjal cultivation is mostly done in the winter season. So, and also BT brinjal, we are saying that it is only for the winter season. BT brinjal was imposed as a target country in Bangladesh under the Agricultural Biotechnology Support Project 2, ABSP 2, particularly after the moratorium in India in 2010, and the court order in the Philippines against BT brinjal in 2013. So after that, our government became very active, and actually the ABSP program, which is funded by USAID and Cornell University in US, they became very active to get it released, at least out of 3 countries of their program. 2 have already said no, so Bangladesh is the only one left. So they became very active, and they started pushing the government. And but Bangladesh environmentalists protested the approval process, and also international scientists protested. And but we were going through a political disturbance at that time, so it was very difficult to talk anything against the government. And government also was very worried whether it will come back after the election in 2014. So the last parliament session was in October 2013, and so they wanted to have it done by then. So when the approval process was completely flawed, I just want to give you a sequence of how they did it. They, after application in August, they took an unusually quick process to approve the BT brinjal. The National Technical Committee for Crop Biotechnology, they got the application in July 2013. But after that, we had writ petition in the High Court, and then expert committee was meeting in September, and just immediately after that, government, discarded the writ petition. Another writ petition was filed on 22nd September, and that had health risk issues. So the court ordered that it should not be released without assuring the health aspects of it. But even that was ignored. An expert committee then sent the file to the National Committee on Biosafety at the Ministry of Environment, and application was approved by, they say that it is, they in fact read only the executive summary of all the documents that they had, and they approved it in October. On 30th October, almost on the last day of the parliament, when the ministers could be in their portfolios, they approved it. No media briefing was done. And Prime Minister Sheikh Hasina was given letters from international scientists and environmentalists. It's like Professor David Schubert of Salk Institute for Biological Studies. He said that unprecedented health hazard to the population of Bangladesh because there has not been adequate safety testing of BT brinjal for human consumption. So there were this kind of warnings given to the Prime Minister, and we made sure that she gets the documents. We supplied it to her office. Finally, on July 13, we get the news in that Bangladesh is set to join the elusive club of 28 GM crop countries, as if it is something to be proud about. And after October, it was approved. And here, as I showed you in the beginning, the different varieties of brinjals which have names of their own. And these also the BT brinjals had, which were converted into BT, also had names. For example, one was called Uttara, now it is BT1. Another was called Kajla, now it is BT2. And Nayantara is BT3. And another one, Uttara ISD 6 is BT4. So this is ISA, they have made a map. So they have taken from all different parts of the country, because we have a diversity in terms of their geographical locations, and only 4 are now given to the farmers. And farmers, from of this area, if they are given the variety from this area, they will not grow. They don't like it because it is not suited to their environment and taste. So, and what happened was, this government came back to power in on January 5th election, but they were in a hurry to distribute the seeds of BT brinjal. And by 5 January, they formed the government, and then by 20th January, they distributed the seed. That means the Ministry of Agriculture, the first thing they did was to distribute the seed. And they were given the seed for first round in 2014 to 20 farmers. This is one of the farmers which received in the first round. But 17 farmers discontinued. In fact, they were so angry that they wanted compensation. And we followed up for the 6 rounds they gave the seeds. And most of the time, we have done some studies in among the farmers they gave. We tried to find out the list of the farmers where the seeds were given. And we found that 68% of the had bad experience. And one important thing was that farmers said that these varieties are unfamiliar to us. And they also did not like the fact that they were not told that what is the name of the brinjal. They don't like this BT1, BT2, and 3. And also they were told initially that they would not require any pesticide, but we found 35 types of pesticides were sprayed at different times for different kinds of insects, including FSB. So you can see some of these pictures here, which were after in the fields. And the complaints were that there were false hopes given to them, and that plants were taking longer time to grow. And they said the flowers which came out were male flowers, no or less fruit. And the losses, because they were doing it in small plots, the losses ranged from Bangladesh taka 15,000 to 30,000. For a small scale farmer, is quite a lot of money. Whereas in the local varieties, for example, this variety called Adgoria, the farmers were earning like 70,000 to 100,000 taka in a season. So they were quite happy with their own varieties. One of the things was approval conditions said that you have to label it with the BT brinjal if you sell in the market. But that was the condition given by the National Committee on Biosafety. But the Director General of the Bangladesh Agriculture Research Institute said it is totally impossible to label the brinjals before going to the market, because they go in big, like the one you can see here, they can't label all these things. So those are actually mixed up with the normal varieties. Now comes the Cornell University issue. Cornell Alliance for Science was launched in 2014, and $5.6 million grant was from Bill and Melinda Gates Foundation to add a stronger voice for science and depolarize the charged debate around the agricultural biotechnology and genetically modified organism. In Bangladesh, they are not even trying to convince us with scientific arguments. They're just making up stories with all lies. You know, they're publishing articles in the newspapers, you know, and not allowing other kind of you know, articles to be published or rejoinders. And, you know, Bangladesh Agriculture Research Institute has now no role of their own to look after their own interest. It is guided by the ABSP tool. That means Monsanto and, you know, the Cornell University. And from the field testing, I can show you the field testing place, you know. They were involved from the beginning. Even the signboards had Cornell University's mentioned here. You can see that all Cornell University was mentioned from the beginning in 2013 in 6 regional field centers, which were started even before, for example, in 2007. So they were doing it without really following any guideline. You can see the fences are broken, people can get in. These are BT brinjal plants inside. So the field testing was not even done scientifically. We have not seen any kind of public document on the field testing also. So the claims of the success stories after repeated failures started coming in, because now have started saying that it's so good that the farmers have started from 20 in 2014. Now 27,000 farmers are growing BT brinjal. Okay, that is true. We tried to find them out in last year. You know, we in different markets, particularly during Ramzan when brinjal is sold at a large scale. And we did not find in many districts that there is any BT brinjal or anything that looked like BT brinjal. So we asked the consumers, and no seller knew about it. None of the consumers knew about it. Nobody heard about it. But this program is, there is one program that is funded by, you know, Cornell Alliance group. Is called Farming Future Bangladesh, and it is continuously, they are having a radio program all the time saying, talking about the success stories, but we never find the actual successful farmers in the field. We have been there, and just let me tell you one interesting thing, that just last month we heard that in one district they gave the seeds to 10 farmers, and we got the list from the Department of Agricultural Extension. So we went there to the farmers and to find out how the plants are growing. 7 out of 10 said they did not even receive the seeds, but their names are recorded as the recipient of BT brinjal seeds. 3 farmers had the seed, but they were afraid to talk without the permission of the Department of Agricultural officials. So now, you know, you can see that how insecure the Cornell University and the promoters are, that they have to come to the Prime Minister and give her a, you know, honor, you know, for her contribution to the development of agriculture sector and ensuring food security. And what is that? Actually, that in response, Prime Minister said that Cornell University has done such a good job for the innovation of BT brinjal. So to hear that, the Cornell University officials came all the way to Bangladesh and gave a crest to Bangladeshi Prime Minister, and to tell everybody that we have the top level support. So don't.
A couple of minutes more.
Okay, I have two more slides. And they even engaged BBC Panorama program. So it was aired on 8 June 2015. They even interviewed me and totally projected out of context. But let me show you that this is Tom Heap, who was the main journalist who interviewed the BT brinjal farmer. And they even used a drone camera, and drone pictures were there. And they even, Tom Heap even ate brinjal at the house of the brinjal farmer and said that he was so happy, 90% success and all this. But this particular farmer, Hafizur Rahman, and where Mark Linus also visited, we also went to his field, and he has had the plants having this kind of BT brinjal, hanging on the plants. So, but when he shows, he only shows the a few that are good. And Hafizur Rahman is not even a farmer. He is a polytechnic graduate and a well-off commercial vegetable farmer. So it's not true that small-scale farmers are motivated and successful. So, and so he was also not very happy. He said that the brinjal has rough surface and gets softened very quickly. So in the market, it does not remain very good. And even the farmers told us that they cannot sell in the village because people see that it does not look very good. So they mix up in the market whatever they have. So BBC finally made a completely false story, and we have complained to BBC, and of course they will not accept it, but that complaint was made by journalists from Bangladesh, and I have myself done that. So anyway, our protest continued in meetings, on the streets, and with all the important people like Dr. Zafrullah Chaudhry, Rizwana Hassan, and we are all environmentalists, they are all there. So, but the intimidation is there. Our farmers, I was briefing the journalist in one of the BT brinjal fields, and afterwards we were attacked when the farmers came and wanted to demonstrate against the BT brinjal. So except few newspapers like New Age and others, other newspapers are not even publishing any of the articles. Bangladeshi scientists who want to develop their capacity in biotechnology and also have a critique of GMOs and particularly of BT brinjal are not allowed to talk. So, but we are hopeful and we think Bangladesh must be GMO free, the world must be GMO free, and we all should work towards a GMO free world. Thank you.
Thank you. Thank you so much, Farida ji. And indeed, not just Bangladesh need to be GMO free, the world needs to be GMO free. And thank you for exposing the lobby of not just the industrial, the agricultural companies, the big act that is pushing GMOs, but the so-called independent journalists and so-called scientists and the various clappers, as we call, or for the GMOs, as Dr. Shiva used to call them. We need to expose even the clappers behind this nexus of those that are masquerading as independent voices to impose these GMOs. Just a reminder, if we have time towards the end, we would take a few questions. For those who would like to, there is a Q&A box in the Zoom chat. So if you would like to have we will try to take a couple of questions towards the end. So please post your questions in the box. And I know we are a little behind time, so I'm going to jump straight to Jonathan. As I said, as we were discussing earlier, in order to circumvent and go around the various bans and strict regulations on GMOs in countries, the industry is now trying to push the new generation of GMOs, the gene editing techniques, as they call them, or the new breeding techniques, as the industry likes to call them. In the UK, for example, the government is proposing to deregulate gene-edited crops and foods so that they would not be classified as genetically modified organisms. And this will have massive repercussions on safety checks, which will be removed, and GMOs will not be labeled. So many scientists have called out the hype and the behind and unmasked the fake science behind these techniques. And I would like to ask if Jonathan could take us through the overall science in this of gene editing and its genetic damage and the off-target effects? And what is this gap between the rhetoric and the actual scientific reality? Over to you, Jonathan.
Right, thanks so much, Ruchi, and welcome everybody. And thanks so much to the previous speakers for that super valuable information. So the talk that I want to give today has two parts. The first part, I will just briefly describe gene editing, which is synonymous with genome editing. Gene editing should be understood as an umbrella term for a basket of techniques. Most involve some kind of targeted cutting system using an enzyme that cuts DNA and which is inserted into living cells. These living cells may be animal or plant or other cells, and although the initial alteration occurs in just one cell, it is important to remember that this one cell becomes the basis for a crop or animal breeding program. The exception to this description is a method called oligo-directed mutagenesis, or ODM. ODM is a technique developed by a company called Cibus, which is based in San Diego. ODM is often referred to as a form of gene editing, but in practice it has virtually nothing in common with other forms of gene editing, since the method involves inserting small stretches of DNA into cells. So no DNA cutting enzyme is used in this case. So it's important to appreciate in the context of this talk that each of these different gene editing techniques likely has a different risk profile in terms of the genetic damage it causes to the genome of whatever cell it is inserted into. It is also important to appreciate that virtually every laboratory or company uses these techniques differently, either slightly or very differently sometimes, or they use different versions of CRISPR, for example, or they use it on different species or at different concentrations, or targets different genome sequences. So in all probability, these differences affect the risk profile of the technique in terms of the genetic damage caused to the genome. So what I'm saying here is that the people are publishing papers describing some of these off-target effects and the various consequences, but we have to understand these as kind of token examples, if you like. So some species methods or genome targets may be much more prone to genetic damage than others or much less. The important fact to keep in mind is that we really have no idea. So when people talk about rolling out gene editing techniques for specialty crops or hard to engineer crops, which they do, these people are venturing far into uncharted realms when it comes to biosafety. There is probably a huge amount of devil in these seeming details, but the bottom line is that we do not know how much these seeming details matter. An important relating issue, and especially this is true for company developers, is that they're not at all transparent about their methods for developing new crops, which they call gene editing. So a case in point is Cibus with its ODM method. This company recently applied to the European Union for approval of the first gene edited crop. And then as the crop began to hit regulatory buffers, Cibus changed its mind and told the EU that its canola was not gene edited after all. It had arisen by chance, they said. So on this basis, and I think this is fairly predictable, transparency is going to be a major issue. Who did what in what crop and so forth. And in the second part of the talk, I'll talk about the steady emergence of reports of genetic damage associated with gene editing. We now know, for example, that there can be major genetic damage and inadvertently introduce DNA sequences at the target site. More slowly, we are learning that there can be genetic damage all across the genome, which can arise for a variety of reasons. And the last thing I want to note is that the word editing itself in this slide is in inverted commas, and I'll use those inverted commas throughout the talk. So these inverted commas emphasize that the term editing is a marketing term. It conceals more than it reveals in that it implies micro changes to the genome and also some similarity with editing on a word processor, which of course is very precise. But the important difference is not only that genome manipulation has nothing in common with word processing kinds of editing, it is that the term editing was invented in advance of anyone collecting evidence for what actually happens inside cells which are edited. So this slide shows you some of the different types of gene editing methods. So as I said, it's a basket of different techniques, and all of them have in common, except for the ODM method, that there is an enzyme that cuts the DNA, and there's a targeting system which brings the enzyme to the place where it's supposed to cut the DNA. This is the theory of the whole thing. And so each one of these methods, the meganuclease, so the acronyms are explained down at the bottom left here. But in each of these cases, there is a protein that cuts and a protein that guides the cutting enzyme to the piece of DNA, to the right piece of DNA. The exception to this is CRISPR. So CRISPR is the bottom right technology, and it uses as its targeting system an RNA molecule instead of a protein molecule. And so this is a small difference, and I'm going to focus on CRISPR for the rest of this talk. And that's because there's most research about CRISPR and its off-target effects and genetic consequences, but also because it seems it's the one that's most used by researchers. It's not necessarily the one that's closest to the market or most used commercially, and that is more of an open question. And we still don't know which kind of technology will win out, as it were, in terms of market forces. But I'm going to focus on CRISPR. And as I said, it has two components. It has a protein component, which is the Cas9 protein. So it's a nuclease. So a nuclease is an enzyme that cuts DNA. And the CRISPR is the RNA component of that. So what a researcher does is they produce an RNA component, part of which is always the same. And then the green part, which is, I should be able to use this little arrow. The green part here is different every time, and that targets the molecule to the genome, the cutting enzyme. So here is a picture of it, a little up close, somebody else's demonstration of how it works. But essentially what you can see here is you have an RNA sequence, which is the gRNA, this part here. And what it does is it basically inserts between two strands of DNA. And when it finds a match that it likes, it cuts the DNA in two, right? And what you have to understand about a DNA cut is it's not an edit, right? An edit would be a replacement of an A with a T or a G with a C, for example, in the genome sequence. But that's not what's happening here. What's happening, and the same is true for all the others, is that simply the enzyme is making a cut. And then what happens once that enzyme is removed and disappears, the cut has to be repaired by the cell. And what the researcher has to do is try to select repair events. So they will basically do this in thousands and tens of thousands, sometimes of cells. They have to select a repair event that suits their needs. So the cell will repair these events, and it does so fairly at random. But it also does it in a kind of a panic mode, because even though this looks like a small change to the DNA, you've just cut it, surely it can just recombine. What you're doing here is you're cutting a chromosome in half, and the chromosome has the potential to when the chromosome is cut in half, that means the cell can no longer participate in cell division. That cell is basically at a dead end because it can't transfer its genome into the next generation in a rigorous kind of logical way, because basically the genome becomes fragmented. So the cell has to repair that sequence, that cut. And so it does so often in kind of a panic mode. So what is emerging from the off-target research is basically that all kinds of strange DNA can be added at the cut site, which is not consistent with a gene editing type description. So one variant of this method is that the researcher can supply their own DNA, which the cell can use to repair the cut. And so what the researcher would then do is basically do the gene editing process, but they would select for repairs that use the double-stranded DNA that they themselves have supplied. So once again, this is kind of a deviation from the whole editing idea in the sense that one can actually introduce, major transgenes and antibiotic resistance genes or herbicide tolerance genes. There's all kinds of traits that potentially can be added that companies are hoping they can just get by calling these gene editing. So this is kind of the state of the market, at least in the US, of gene editing. From probably a year ago, I made this slide. So I just wanted to give you a kind of overview of where we're at. So in this first example, Penn State University has made a non-browning mushroom. And they published one paper, and there's no sign that will ever come to the market. And then the other examples are made by companies. And the reason I put boxes with question marks inside them is that we don't even know enough about them to have photographs available. So practically no information is available about these. And then in the case of Cibus's canola, they consider they're very close to the market. But as I said, there's been some interesting things going on with their approval process. Right? They basically changed their minds that their crop is a gene edited crop. And this underlines the whole issue of transparency. No one can really know what the company has done to its crop if they don't honestly tell us. So I just want to go through some of the research about the kinds of imprecision that are associated with gene editing. So this was a paper that was published a few years back, I think it was 2016, and it's kind of kick-started the whole idea that gene editing might actually, despite its name, not be very precise after all. So there were some papers published before this, but the research community kind of felt that they could ignore that because the authors of the research were not famous, and there were calls for their papers to be retracted as they couldn't possibly be true, and so on and so forth. But eventually, with the publication of this paper, it became acceptable in the scientific literature to argue that there was imprecision at gene editing sites. And what these people showed is that the gene edited cell is basically repairing the breaks produced by the gene editing mechanism by leading to either large losses of DNA or to complex rearrangements of chromosomes, which are also highly problematic events for the cell. And so they're likely to lead to basically mutant plants. So, and then there's the whole issue of how does a researcher actually make the edited crop in the first place? So one of the key issues that nobody seems to be paying too much attention to is the fact that in order to make an edited cell, you have to introduce the editing apparatus into that cell. And the way that most people have been doing this is by basically shooting DNA, introducing DNA into the edited cells, which codes for the enzymes or the RNA or the targeting protein that does the editing process. But what that means is that you have to introduce a lot of foreign DNA into those cells. So you can't just add the DNA that codes for the CRISPR and the Cas9. You also have to add all these ancillary parts, which potentially can end up in the genome, very likely, in fact, can end up in the genome of the edited crop. So instantly we have a problem here in that the edited crop is going to be potentially have all this foreign DNA, which comes from all over the biological world, put into it. So this is an issue of the source of the DNA. Then the next question is, you have to prepare, and I'm giving an example here of plant cells, but similar comments could be made about animal cell editing, is that you have to prepare the plant cells to receive the DNA. So you're going to shoot the DNA. You're going to use basically a modified gun to shoot the DNA on pellets into the cells, and you have to get the cells in a fit state to receive that DNA. And the way that you do that is basically put them through a thing called tissue culture, which is very mutagenic, right? So thousands probably of mutations in each cell arise as a consequence of this tissue culture. So, one of the ironies of all this is that the editing process is supposed to be very, very precise, but the developers are putting their cells through extremely challenging situations. And likely the number of mutations that will be introduced by the editing process will be dwarfed by the number of mutations introduced by the kind of procedures, what you might call the ancillary procedures that go along with this. So let me just show you an example of this, right? So imagine what happens when you want to shoot a pellet. So I've got it drawn this as a red pellet with some orangey colored DNA on it. Essentially, you put the DNA on the pellet, and then you're going to shoot that using a gun into the cell. When you do that, the pellet basically has to pass through half of the cell before it gets to the nucleus. It may pass through a chloroplast or a mitochondrion, some cellular organ. And when it passes through those organs, it collects DNA. They're also full of DNA. So it will collect pieces of DNA. It will lose pieces of DNA. It will finally lodge in the nucleus. And in order for the DNA that you've added to insert into the nucleus, you first have to damage the DNA. So the pellet is basically crashing into the chromosomes in the nucleus and damaging them. And we know this because if you don't damage the DNA, then there's no reason for the cell to take up the DNA introduced in the pellet. So you've got this very damaging process of shooting DNA in pellets into the nucleus that also results in major genetic damage. So this isn't the only method of doing it, but it is the most popular one. And so I'm just focusing on that really as an example.
Jonathan, if I can request you to wrap up in a few minutes, couple of minutes.
Yeah, we're nearly there, Ruchi, thanks. So the third kind of genetic damage that interests me, and what I'm doing here is picking and choosing bits from all over, but essentially simply because there's not time in this talk to talk about all the different kinds of genetic damage. But this is a really interesting one. So this is a study done last year where researchers basically took, they edited some mouse cells, and they took those mouse cells and they then sequenced the DNA of the insertion sites. And what they discovered is that they had inadvertently added DNA from E. coli, which is a bacterium, DNA from a cow, which is bovine, and they'd also introduced DNA from goats. And they scratched their heads for quite a while, like, where does all this DNA come from that we didn't intend to put into these cells? And it's now ended up in these cells at the editing site and at different places in the genome. And it turned out that they're contaminants of the process used. So basically, as I said, they're taking DNA and they're having to purify some DNA in order to shoot the editing proteins shoot the editing mechanism into the cell, but that DNA has contaminants in it. And the contaminant in this case was E. coli because they'd grown up the DNA in an E. coli cell. And the bovine DNA and the goat DNA came from other forms of contamination that were basically requirements of cell culture, right? So cell culture requires purified proteins, and those purified proteins come from cells that contain DNA. And if you use bovine DNA or goat DNA, that is where your contamination comes from. So this is completely unexpected, and it is totally inconsistent with the whole idea of genome editing. So this is really a summary slide. So we're just about at the end, Ruchi. So what I want to really say is that genetic consequences stem from the manipulation of cell cultures and the need to introduce the gene editing components into cells, right? So this is basically all the ancillary stuff that you need to do your editing in the first place. And the damage to the genome is essentially random. It's not going to occur at your edited site. And then there's another set of genetic consequences which stem from the functioning of the edited gene editing machinery itself. So the gene editing machinery can cut the DNA on the target, but it can also mistakenly cut places in other places in the genome, which people call off-target cuts. And I haven't even had time to talk about those, but basically it's not a guarantee that the editing machinery will only cut where you want it to. And all of these, if you add up all these issues, what they basically do is they say, this places a very high premium on the diligence and competence of the breeder, right? Because all these changes can happen, but it's up to them to make sure that those don't find their way into the final product, that they exclude ones, that they actually even know how to detect these changes in the first place. And at the moment, I don't know how many people have followed the hornless cow story, in which basically a company was pioneering cow breeding with breeding hornless cows. They basically inadvertently introduced an antibiotic resistance gene into their cows. And they had no idea it was there. It was perfectly obvious. Just a simple piece of sequencing from the FDA detected it, but the company itself didn't do that, right? So there's a huge question over the diligence and competence of all these startup companies and the multinationals that are actually doing this. So I'm going to end there, and I'm happy to take questions if there's time, but that's up to you, Ruchi. So thanks very much, everyone. Oh, please subscribe to our website. Given the way the internet is these days, it's going to be difficult for you to keep up with all this information if you don't subscribe to us. So thank you.
Thank you, Jonathan. I know it's not fair to ask you a huge, such a large amount of information and the science behind this in 15 minutes. And we promise to keep following up with this. And as Jonathan said, you can subscribe to, I think you've given us the link again, and we will add links of the organizations and the participants that have, panelists that have participated today, as well as we will be linking the presentations on our website after the webinar today. Thanks again, Jonathan, and we will try to come to questions. I see a few already after we finish with all the panelists. As I said, there is a massive imposition, an attempt to impose these so-called new breeding techniques in many countries of the world. There is a process right now happening in UK, which is trying to propose to deregulate these gene-edited crops and foods. And if you look at the GM Watch website, they have requested people to participate in this process and let the government know about your views. So check out the website for GM Watch. In Italy, for example, in January, a coalition of environmental organizations, farmers organizations, and movements managed to stop the government for pushing 4 bills that would have deregulated and allowed the entry not just of the new GMOs, but also of the old GMOs. And they were able to stop thanks to a massive social media, as well as overall campaign and an alliance of various actors coming together. Similarly, thanks to the work of farmers organizations and environmentalists over the years, you have seen in Mexico early this year, a presidential decree was given to begin to phase out from both glyphosate and GM maize. And this decree would ban both the use of GM maize in the field and in food in order to protect the health and biodiversity. From Adelita, if we can understand what exactly is happening in Mexico, what does this decree really mean for the movement for GMO-free and seed sovereignty and food sovereignty in Mexico? If I can come to Adelita. Thank you. And Carla, my colleague, will translate Adelita from Spanish to English. Thanks.
Thank you very much, Ruchi. And estoy muy contenta de estar con ustedes y agradecida de esta conferencia y de escuchar la participación de los compañeros de Bangladesh y esta última explicando tan claramente este tema de la edición genética.
I'm very happy to be here with all of you, and I'm very happy to hear all the different opinions and different situations that are going on in Bangladesh and the Philippines, and especially for the recent very detailed presentation of the threats of gene editing.
A mí me gustaría empezar señalando que México es uno de los 8 centros de origen, incluido en Mesoamérica, de la agricultura.
I would like to start by signaling that Mexico is one of the many centers of origin of agriculture in the world.
A mí me gustaría platicar esta larga historia de la protección del maíz en México, que inicia con pues esta situación de México como un centro de origen de la agricultura.
So I'd like to share the long history of the protection of maize in Mexico, and the point of origin of the story is the fact that Mexico is one of the centers of biodiversity, original biodiversity in the world.
Y para nosotros, para la gente que vivimos en México, en Mesoamérica, el maíz tiene una importancia central en nuestra cultura. Se dice que el maíz nos hizo y a la vez nosotros hicimos al maíz.
And for all of us living in Latin America, the corn plant is something central to our culture. There is a saying that the corn plant made us just as much as we created the corn plant.
Porque en su conformación como maíz y en su evolución desde el teocintle, hubo la necesidad de la mano del hombre.
Because in the evolution of corn, from its origins as teocinte, human hand was very necessary in its growth, expansion, and evolution.
Y podemos ver siempre al maíz expresado en todos los murales, códices, etc.
You can always see maize expressed in our artistic works, in our murals, in all of its glory.
Entonces, hablamos que el maíz tiene una historia de 8,000 años de éxito, de mejoramiento y progreso.
So we're talking about a crop that has undergone 8,000 years of evolution and betterment and progress.
Es un cultivo altamente adaptable a cualquier altitud. Lo podemos encontrar desde Argentina hasta Rusia, desde África hasta Estados Unidos y a diferentes alturas, desde el nivel del mar hasta 3,000 metros sobre el nivel del mar.
It is a crop that is extremely adaptable to different temperature ranges and in particular different altitudes. And you see this expressed as you can grow corn anywhere from Argentina to Russia or to Africa to the United States. You can grow it from at the 0 degrees of sea level or to 3,000 meters above sea level.
En esta gráfica podemos ver la gran adaptación de algunas de las razas de maíz.
In this graphic, you can see the great variety of adaptation found in maize.
Y esto lo hace ser el cultivo ideal para el cambio climático.
And this makes it be the ideal crop for the different changes coming with climate change.
En la actualidad, el maíz es el cultivo con mayor volumen de producción en el mundo, superando incluso al trigo y al arroz.
Today, the corn crop is the largest produced crop in terms of volume, and it even supersedes rice and wheat.
Por supuesto, Estados Unidos es el mayor productor de maíz y de maíz transgénico.
The United States is the largest producer of corn and also the largest producer of GM corn.
Y su gran difusión se debe a la gran diversidad de usos. Podemos encontrar maíz en todos estos productos, convertido en azúcar, convertido en alimento para ganado, en textiles, en biocombustibles, etc.
And there's a great diversity of the use of corn and corn products as well. You can find it in all sorts of things, from processed sugar to biofuels to animal feed to different sorts of oils and food additives.
Y dadas estas características, esta importancia y el gran significado que tiene para la gente en América, no solo en Latinoamérica, en toda América, pensamos que el maíz es un grano en disputa.
And considering all of these different characteristics and its importance to not just the Latin American people, but all American people, we can consider that corn is a grain in dispute.
Y por eso desde hace muchos años en la campaña Sin Maíz No Hay País, que es un gran movimiento que agrupa a más de 300 organizaciones.
And this is why we've now come together as a campaign of Without Maize, There's No Country, which is a group organized by over 300 different organizations.
En esta campaña decidimos tomar la ley en nuestras manos, dejar de pedirle al gobierno que no aceptara el maíz transgénico, y tomamos los instrumentos jurídicos para defender al maíz y nuestra alimentación.
So with this campaign, we decided to take the law into our own hands and stop asking the government directly to outlaw GMO maize. And instead, we decided to take the tools of the justice system into our own hands and take action on our own.
De esta manera, presentamos una demanda colectiva, a class action, para defender la biodiversidad del maíz.
So together we presented a class action lawsuit to defend the biodiversity of maize.
Y la gran noticia fue que un juez nos concedió una medida cautelar porque nosotros justificamos que la contaminación era tan fuerte que podría contaminarse todo el maíz en México y que se debían suspender cualquier permiso esto fue desde 2013.
And the great news is that a judge listened to our plea and did approve a move to start the banning of GM maize, because we showed that the contamination of GM maize was so profound that it was harming our natural biodiversity. And this movement started in 2013.
Desde esa fecha, la siembra de maíz transgénico está suspendida porque el juicio todavía se lleva adelante, con más de 150 impugnaciones.
And since that date, the approval and the usage of GM maize has been suspended in the country, especially since more than 150 violations have been found.
Y en el 1 de julio de 2018 tuvimos un cambio de presidente, un presidente que llegó con mucha aceptación popular y se comprometió a prohibir el maíz transgénico en México.
And since July 2018, we've had a change in presidency, and he's a president that came in with a lot of public support, who promised to prohibit the use of GMO maize in the country.
Entonces, en el 2019, la Secretaría de Medio Ambiente negó la importación de glifosato para empezar. pues había evidencia de la presencia de glifosato en orina de niños y en agricultores.
So in 2019, the Environmental Agency of Mexico prohibited the importation of glyphosate, especially after glyphosate was found in the urine of children and of Jalisco and in different farmers' urine as well.
La justificación fue el principio de precaución.
The justification was the precautionary principle.
Exacto, simplemente. Y un año después se han negado 76 solicitudes a 15 empresas que equivalen a 132.344 toneladas de glifosato.
And since then, 76 permits have been denied, and that represents 15 different businesses, which amounts to 132,344 tons of glyphosate.
Por supuesto, México no produce glifosato, todo lo importa.
Of course, Mexico doesn't produce any glyphosate. Everything is imported.
Este decreto prohíbe en un tiempo, en un periodo de aquí al 2024, el glifosato y el maíz transgénico, tanto para siembra como para consumo.
So this decree prohibits the importation and the use of GM maize and glyphosate with a phase-out period to 2024. And this includes the planting of GM maize and also GM maize found in any food products.
Ahora tenemos el gran reto de implementar este decreto con múltiples acciones y sobre todo frente a una gran oposición en el país de los empresarios agropecuarios que son muy poderosos.
And now we face the challenge of actually implementing this decree, especially since we're facing a lot of opposition from very powerful actors in the industrial agriculture industry in the country.
Y bueno, finalmente yo quería mostrarles a ustedes una lámina que representa parte de lo que hemos hecho en este movimiento, que es un gran tapete de maíz que hicimos en la gran plaza que tenemos en la Ciudad de México, un gran tapete que dice México no quiere al maíz transgénico y sobre todo, si quieres traducir, Carla.
And lastly, I would have liked to have shown you a slide of a sort of mosaic that we made out of different varieties of traditional corn, saying Mexico doesn't want GM corn.
Y con esto también resaltar que pues este ha sido un movimiento de muchos años y de muchas personas para lograr parar en México la siembra de maíz transgénico.
And lastly, to highlight that this has been a movement of many different actors and organizations over a long period of time to accomplish this banning of GM maize and glyphosate in the country.
Muchas gracias.
Thank you very much.
Thank you, Adita. Thank you, Carla. Indeed, yes, the decree to phase out glyphosate is a welcome step, as well as the banning of GMOs. The challenge lies in the implementation of these decrees and to resist the continuous pressure from the agricultural lobbies, agribusiness lobbies. The governments actually, farmers, movements, seed savers throughout the world are already practicing a poison-free, GMO-free agriculture. So it's time that governments catch up to the small farmers and the women that have been practicing this ecological agriculture for millennia. Without taking any more time, if I can now invite Dr. Vandana Shiva. Dr. Shiva, you have been battling GMOs and the lies of the industrial agriculture and the agribusiness for decades now. Tanya was started in response to the attempts by the biotech industry to take over the seed. Could you tell us, could you take us through, could you tie up all this, could you take us through these decades of false promises and lies? What do the future threats look like and our resistance to it? Thank you.
Thank you, Ruchi. Thank you to Farida for following our beloved Brinjal all these years. In 2010, India had the ultimate democratic test. 7 public hearings were organized across the country to listen to scientists and farmers and consumers about BT Brinjal. And we put a moratorium, and Gates ran with it to Bangladesh. 2000 is when they first introduced the idea of golden rice. Of course, they put in front a few scientists, but there were 80 patents associated with golden rice. And I wrote then a piece called Blind Approach to Blindness Prevention. As Chito so clearly pointed out, we have so many alternatives to have vitamin A-rich foods. But what the golden rice and the eggplant shows us is we are not dealing with science anymore. Science is what tells you the eggplant has failed. Science is what tells you the GMO golden rice will not deliver. It is a propaganda machine with the hub being the Cornell Alliance of Science that is running the system they claim to be science. But that's not new. You know, it began with Bacon, who said everything of the past we are going to call effeminate. And I'm going to give birth to a masculine birth of science, where nature is subjugated, where other cultures are basically declared ignorant. And I'll build a super race of superheroes, and we will decide what reaches the public and what will not. So if you read Atlantis and what's going on today, you realize there's been a few hundred years in the making. And in a way, all that we see is the current version with the new technologies. Thank you so much, Jonathan, for walking us through the continued fraud on genetic determinism. And Adelita, dear Adelita, I am so happy to have been part of your defense of maize, having come back again and again and again to Mexico. And we'll be together for the brinjal and for the rice. You know, in India, we call rice akshat. As Dr. Risharia taught me, we evolved 200,000 rice varieties. India is the Vavilov center of the indica varieties of rice from one grass, 200,000 varieties. Akshat means that which is not broken. Shat is the broken. Golden rice is the broken rice, which is breaking other systems, breaking democracy, breaking science. So if I look back, you know, this is not my field. I was trained in quantum theory. I did a PhD in non-separation and non-locality. But it was 1984 in the Punjab protests of farmers that forced me to look at what happened, because the Green Revolution was given a Nobel Peace Prize. So why is there violence in Punjab? And the reality and the narrative didn't hold together. It's very interesting. Bill Gates' new book on climate change celebrates waterlog and the Green Revolution, which devastated Punjab and is still at the roots of the current farmers' protest. He, says all of us are eating because waterlog created semi-dwarf varieties. No, people are starving because of the Green Revolution. And then he goes on about synthetic fertilizers being a climate solution when we know they're a big part of greenhouse gas emissions, nitrous oxide. So what I realized when I studied the Green Revolution is scientifically and through indigenous culture, you look at systems as a whole. And so you evolve systems of biodiversity that work in cooperation with each other. In India, the Navdanyas and the Baranajas and the Satnajas. And in Mexico, the Milpas. Never a corn alone. Corn with the squash and the beans. And together, they make the production even higher, the three sisters. So the first problem with this paradigm that continues is violence and war. It's a mentality of war. And when I look at the Gates and the billionaires and the deregulation and the gene editing, I say all that has changed is the cowboys don't ride horses anymore. They want to ride on life. And all indigenous cultures, all biodiversity, all the richness of the earth is now the new frontier of the Wild West. So beginning with the colonization and the creation of frontiers, the Green Revolution was a continuation of Hitler's wars through chemical warfare. And Rachel Carson wrote about this. Sir Albert Howard, in his book, Agricultural Testament, wrote about it. I call this the monoculture of the mind, where you take systems with diversity, you reduce them to one crop, a rice or a wheat in monoculture. And then you take a part of that crop, the grain, and you destroy the straw. As the scientists who founded it, Swaminathan Ji has admitted, they changed the partition. They didn't increase the yield. They merely put more in the grain and pumped huge amounts of chemicals. And the reason they had to make the dwarf varieties is our native varieties refuse chemicals. So they adapted the plant to chemical warfare. And then they created a very reductionist term called yield. And Gates's new book is only about yield, the yield gap, the yield, meaningless term. Yield doesn't tell you what's the state of the land, doesn't tell you what's the state of the farmer, doesn't tell you what's the total production, the total output of the farm, doesn't tell you what's the quality of what grew. And as our work is showing, the indigenous seeds have far more nutrition and definitely more quality, which is why people are turning to heirloom tomatoes and heirloom carrots, and because they have far more phytochemicals that are good for taste and good for health. So over these years, I said not reductionism and yield per acre, but we'll mention nutrition per acre and wealth per acre and health per acre. And we could feed 2 times the world's population if we were honest in our measurements. So not only is there a propaganda machine, there's a mindset, a reductionist mindset, a monoculture mindset. And then there's a monoculture mindset of reductionism based on determinism. Living systems are self-organized systems in evolution. Genetic reductionism first begins by treating living systems as machines that are put together externally by the genetic engineer. And that's which is the ground of the idea of patenting. I got into seed saving and fighting patents and fighting GMOs largely because of a meeting in 1987 where the corporation said, we now have to claim we've invented the seed. I said, but you haven't. The seed makes itself. And since 87 till now, we have been dealing with this lie, and thank goodness most countries don't accept this lie. At that meeting, they said very clearly, we have to have GMOs in order to have patents. Even the new gene editing that Jonathan worked us through is being pushed by Gates because he already has huge investments through Editas, a company totally for patenting gene-edited products. So it is still about patenting. And these are new GMOs because GMO means genetically modified. When you scramble the amazing complexity of genomic self-organization, it's a genetic modification. This is why we had biosafety laws. And I was part of the founding group for the United Nations to write the framework for the Convention on Biosafety, for the Protocol on Biosafety under the Convention on Biodiversity. So it isn't the case that we did not correct the damages of the Green Revolution. We created not just agroecology, but the FAO led the entire articulation. And now FAO has been hijacked by Bill Gates. And I would call the New York Summit the Gates Summit of anti-food. Because for me, golden rice and impossible burger and cellular production of meat is fake food. So here again, in terms of food, we are up against fakeness and untruth versus the truth of good health, eating real food. So the new GMOs should be regulated under biosafety, and the European Court of Justice has said they are GMOs. So the rush to push gene editing is to undo the laws on biosafety regulation, but also because gene editing, the same tools are used to do digital genomic mapping, undoing the laws for protecting the biodiversity sovereignty of indigenous communities and third world countries, which are biodiversity rich. This is what led to the Convention on Biological Diversity. Under the Convention on Biodiversity, it led to the Nagoya Protocol, and Adelita is a major leader in protecting and defending that protocol. It led to the Seed Treaty. Now, Gates to the Global Empire report has the details from Esquinas, who was the architect of the Seed Treaty at FAO. Adelita has talked about the Nagoya Protocol. So when I talk about the Wild West, I talk about the fact that it's not that indigenous people didn't have laws. They had the law of Mother Earth. They had natural law. They had most amazing laws that for thousands and thousands and thousands of years, they could live on Turtle Island and not ruin the land. And those laws were broken by the so-called cowboys of that time, who treated the occupation as the conquest of the Wild West. The conquest of today is into the interior spaces of life. And as scientists like Jonathan remind us all the time, just as in a mixed field of biodiversity, of Navdanya, of Milpa, there is amazing self-organization at the systems level. Within every living organism, the principle is self-organized complexity. This is established. This is established scientifically. And when you shoot a gene, either through the old GMOs or shoot a protein in the new GMOs, as Jonathan explained, the equivalent is shooting a bullet. And the lie is claiming that when you shoot a man dead, you gave birth to him. Scrambling the tree of life without knowing what you're doing with all the examples that Jonathan gave us is really a crime against living systems. It was treated as a crime under the biosafety laws. So we are living through a period where the billionaires like Gates and the companies, their investment funds like BlackRock and Vanguard and State Street, and my book Oneness versus 1% goes through this, because I realized, one, that in Paris at the climate summit, Gates had emerged more powerful than the governments. So they were not regulating and he was regulating them. And second, that buyer bought Monsanto, so something had shifted. Why? It was like musical chairs. So what you have is billionaires, because of deregulation, wanting no regulation. And he calls this, if you see the word he uses most, is zero. He's the one behind zero budget economics, no liability, zero budget farming to destroy ecological agriculture. Tried it in Andhra Pradesh. So here's a zero mind pushing a zero agenda to try and annihilate the work and bring it to zero. And our work is to protect the Brinjal, to protect the rice, to protect our milpa, to protect knowledge, to protect science, and together to protect democracy. What we are living through is an attempt to destroy knowledge and democracy irreversibly. Our work is to continue the work we've been doing with new vigor, new solidarity, and I want to thank you all for being here together.
Thank you. Thank you, Dr. Shiva, and thank you all indeed. We have a couple of questions, if we could take some. We are a little behind, quite behind on time, so I'll try to take a couple of questions. There's one problem for Jonathan. As a longtime activist, can you spell out the various new gene editing names and types to watch out for, CBUS and other names and definitions for us to use and share and to educate others? and a copy of links. We could share some links together if there are additional links from all the panelists. We will anyway share that if there's a quick answer.
Yeah, I'm happy to share some of those. I can put up some links. You know, there's a company, Cebus. There's also another company called Calyxt. But there's also, you know, one thing that's interesting about all this is that there are some, you know, what you tend to hear about are very small companies. You know, these are kind of startups. But the real people behind this who really want to use this is Dow and Bayer and the bigger companies, right? They're kind of waiting in the wings, using these small companies as front men, if you like. And so, you know, we should know the names of these little companies, but we should also understand that it's the big companies that really are behind all this. You know, they have the lobbying power to make the governments change regulations and to deregulate GMOs and so forth. You know, this is not a technology that's going to be controlled by the little guy. And so you also hear people talk a lot about how this will be done in garages and so forth, right? There's no chance that you know, companies that set up in garages are going to control the food supply, right? Or offer, you know, important products for the food supply, because these people don't have the marketing now, they don't have the testing now, they don't have the breeding, they don't have the breeding contacts and so forth, right? This is not going to happen in garages. This whole idea of startups in garages is just a front that's put up for the benefit of the media and for the benefit of populations. So I can share some contacts, but you know, for the most part, the companies that are involved is Dow and Bayer and you know, the real big companies. But you know, the most useful thing I can offer really is to go to our Independent Science News website and look at some of the articles. There's not the articles are not too long and there are not too many, but they kind of summarize a lot of the issues of gene editing in terms of genetic damage, but also in terms of the paradigm that Vandana was talking about. You know, the idea that you can, we published an article, The 3 Myths of Gene Editing, which is basically about the idea that if organisms are self-organizing systems, whether they will be amenable to gene editing or not, whether they're amenable, what does this have to do with the failure of GMOs as a concept, right? You know, we've seen all the history of GMOs has been the companies have this tiny limited number of products, right? They have their herbicide tolerance and they have their insect resistance, but they never seem to come up with the, you know, no reliance on fertilizers, no reliance, you know, the resistance to climate change. They never come up with the complex traits, right? And it has to do with the conceptual flaw that the companies have, which is that they have a genetic determinist mindset, and that mindset basically is a road to nowhere. And so this is the fundamental issue here. So I've kind of deviated a bit from your question, but I just wanted to add that to kind of supplement what Vandana said. But thanks for the question.
Thanks, Jonathan. I will quickly combine one or two more. Will the move to ban glyphosate and GM corn in Mexico be affected or hindered by trade agreements in place between the US, Canada, and Mexico? Adelita, if you have a quick answer. Is Adelita still there?
Sí, aquí estoy. Carla, la pregunta es de si los tratados tienen una influencia.
Sí, cualquiera de los tratados, en particular
Sí.
NAFTA, puede prohibir.
Por supuesto. El Tratado de Libre Comercio ha sido muy determinante en la política agroalimentaria de México. En los 30 últimos años tenemos importación de maíz. De hecho, importamos 30% de la demanda de maíz de Estados Unidos. Entonces,
That's correct. The free trade agreement has determined a lot of the agricultural policy in Mexico, especially in terms of importation. And this is considering that over 30% of corn consumed in Mexico is imported.
Sí. Esto es muy relevante para el gobierno actual. Está tratando de cambiar esta política con mucho más apoyo a la pequeña producción campesina, a la producción de pequeña escala y dejar de apoyar a los grandes productores, intentando que el mexicano o la mexicana no tenga que emigrar a Estados Unidos.
This has been very relevant for the current government, and they are actively trying to change this amount of influence in our agricultural policy, and in particular trying to shift the emphasis away from large corporations to instead start to support the small farmer, especially so those farmers can stop having to emigrate to the United States in order to have a livelihood.
Es el gran reto que tenemos de aquí al 2024. Entonces, pues esperemos que lograrlo y que México vuelva a ser, como lo había sido en muchos años, autosuficiente en la producción de maíz.
This is the great challenge that we face from now until 2024. And we're hopeful that we can return to the state that Mexico is in prior to these free trade agreements, to being fully self-sufficient and being fully sovereign in our food.
Gracias.
Thank you.
Thank you, Adelita. I will just combine a few questions because we are really out of time, and I know it's very late already in Philippines and Bangladesh. One question: how do you bring youth into the movement, and what advice do you have for small farmers? And are we too late or too weak to oppose this colonization of our health and bodies through GMOs? Dr. Shiva, would you take that?
Yeah, very quickly. You are never too late to do the right action. You never have an excuse to say it's too late. In fact, the more urgent things get, the more important it becomes to act. And what are we talking about? We're talking about the food we grow and eat. If we are willing to give up our very being and allow our bodies to be messed up even more by fake food than they have been by ultra-processed food, and the WTO rules of pseudo-hygiene in the sanitary and phytosanitary agreement, you can be absolutely sure this will be, for a while, a planet of very sick people, where the same people who do the genetic engineering own the pharmaceutical industry and make money both ways. You know, buyer, after all, is big pharma. So we have a convergence of big tech, big biotech, big ag, and big pharma, and big media, all of it together. So I believe as human beings, we have a duty to ourselves to protect our health and stay free, and not allow our food sovereignty and food democracy to be hijacked. As far as the youth are concerned, you know, all of us began our journeys as youth. There is no ready-made place. You grow by learning. Webinars like this are places where the young people can be involved. Finding out more, if you're interested in truth about genetics, become an intern with Jonathan. You want to know about golden rice and rice? Take help of Masipag, Farida, and Ubenik, and of course, Mexico. Our possibilities are limitless of where you begin and where you learn. But I can tell you, a lifetime of activism, learning never gets over. And there's no perfect age. There's no perfect person. Each of us owes it to the earth, owes it to our ancestors, owes it to ourselves, owes it to future generations, to not let a gang of 5 destroy our world. We have to do more for this beautiful earth, her biodiversity, who are our relatives, for all the amazing farmers, including those who are protesting in India, and everyone's health. the lies about health, the lies about agriculture, the lies about biodiversity. You know, we have a treaty, and I would request Adelita to get her government to notice this. And we need an alternative response. We got a treaty for Convention on Biological Diversity. And it created the IPBES, the Intergovernmental Panel on Biodiversity and Ecological Services. And suddenly, the World Economic Forum and the Brexit, England, have put together the Das Gupta Review on the economics of biodiversity. And the economics of biodiversity is put it on portfolios for speculation and making money. Make it a financial asset. No, everything that was reduced to a financial asset was destroyed, beginning with the subprime crisis. Beginning with the issue of food, when it was put, the whole hunger epidemic grew after food was made an asset after the 2008 collapse. So we've got to bring life and biodiversity back into the web of life. We've got to bring our rights as human beings back into the rights of Mother Earth. It is the most foundational democratic struggle we have to create. And the divisions that have been created between us, we must constantly transcend them. It says that division was manufactured to make a divide and rule by the rulers. We will not be divided. We are one humanity on one planet. Lots of love, lots of strength. You know, when I started to save seeds, Monsanto and the buyers and the Ciba-Gaijis were saying, by the year 2000, there will be no non-GM seed. By the year 2000, every farmer will have to buy seed every year from us. We'll be 5 companies. Look at the world around you. Yes, the 5 companies are there, but most seed is GMO free. Seed saving has become an activity in the north and in the south. Seed libraries are going, community seed banks are growing. Marcy Bagh has done such amazing work. Navdani has created 150 community seed banks. We didn't let them get away with that vision of only GMOs. We shouldn't let the billionaires get away with only fake food, only golden rice, only BT eggplant, only our knowledge, our fake knowledge, only our fake knowledge, fake food, fake democracy, fake this and fake that. It's really about truth versus this fakeness. And we come from a civilization where we think of truth as really the ultimate reality. That's what Gandhi fought for. And we call it, working for it, we call it Satyagraha, the force of truth. And we work with the belief that because we are working in the long arch, we are working in alignment with biodiversity, life on Earth, and the rights of all human beings. Satyameva Jayate. The truth wins in the long run. In the short run, these rascals look like they're powerful, but they work with empty power, because bullying power is not true power. Destructive power is not true power. Violent power is not true power. Non-violent, creative solidarity, cooperation, mutuality, that's our strength. That's the strength of nature. Let's build on it. Let's have a dream that 10 years from now, these games will be over, like we saw in the 30 years of seed saving. Monsanto disappear and Monsanto's dream disappear. We've got to save real seed, real food, real democracy. Thank you.
Thank you. Thank you so much, Dr. Shiva. And on that note, I will thank all our panelists. I'm afraid we cannot take any more questions. We are completely out of time. And as Dr. Shiva, we are all in this together. Please join campaigns. Navdanya will be launching a call to action if you would like to join us in our work to transition towards biodiversity-based food systems to live in harmony with nature. You can write to us at info@navdanyainternational.org. We will write everybody's emails and organization details after this webinar to all of you. Once again, thank you very much, Chito. Thank you very much, Farida ji. Thank you, Adelita. Thank you, Jonathan. And thank you, Dr. Shiva. And thanks to all of you for being here.